Classical chemotherapy developed mostly by screening procedures to see if one could find poisons that killed cancer cells and not normal cells. With the advent of antibodies that could specifically bind to proteins, antigen, the cell surface, it became possible to make anti-cancer agents, drugs, that just targeted or mainly targeted cancer cells. And we explored many proteins, but what we learned is that we had to have a target that was on the cancer cell but could not be on any essential normal cells like liver, kidney, brain, so on. So the best target turned out to be what we call lineage-restricted differentiation antigen. An antigen that's in a normal B cell and in B cell malignancies but in no other cells. So if you kill the malignancy and the normal B cell, the normal B cell could regenerate from the stem cell, but you wouldn't damage the person. But instead of taking a piece of a toxin we purified in an antibody and chemically conjugating together, we used DNA gene splicing techniques to just use the essential part of the antibody and the essential part of the toxin and make them all together in one molecule, throw everything else away. And so that was our approach. The biggest early success was with BL22, which as an antibody fragment that reacts with CD22 in B-cell malignancies. And Dr. Kreitman in clinical trials showed that BL22 could produce about a 50% complete response rate in patients who'd failed all therapy, so-called refractory chemotherapy-resistant hairy cell lymphoma. But unfortunately, BL22 did not work well in more common B-cell malignancies, like chronic lymphocytic leukemia or acute lymphoblastic leukemia in children. So in the lab, we overcame this deficiency by using techniques to make a higher affinity antibody, an antibody that bound much better. And that new recombinant immunotoxin we named HA22, the high affinity 22. And that is now licensed to MedImmune. It's being developed by them and with us under a collaboration agreement. But in patients with mesothelioma whom we're treating with immunotoxin SS1P, we are seeing some success, some responses, but we can only give one cycle of treatment because their immune system makes antibodies. So our major effort in the lab is to try to make new immunotoxins that are not immunogenic by removing what we call the T-cell epitopes, removing certain amino acids that promote immunogenicity. So one major effort will be to continue to make less and less immunogenic immunotoxins so we can give more and more cycles.